Bismillahirrahmanirrahim. Today we are going to discuss microbial tests of meat. Meat is not a sterile product. Meat regularly contains different type of bacteria. Most of these bacteria are harmless, but many meat products are associated with harmful or pathogenic bacteria such as Salmonella, pathogenic Escherichia coli, Campylobacter, Listeria, and others. Detecting the different pathogens usually relies on using different type of selective media. These are the media that allow only certain type of bacteria to grow. This may be due to addition of different sugars or other substrates or chemicals that inhibit or kill different type of bacteria. An effective selective media will select for the bacteria of interest by allowing the target bacteria to proliferate while inhibiting the growth of all or most of other bacteria. Two protocols we are going to discuss are basically two examples of how selective media can be used to detect the presence of undesirable bacteria. There are countless type of selective media available and different type of bacteria for different type of bacteria, but the principle we are going to discuss ahead will remain same almost for all of them. The first is coliform test. Coliform are the bacteria that is generally found in intestinal system of animals and in turn in animal fecal material. So the presence of coliforms in meat sample is an indication of fecal contamination. The coliform test described here uses violet red bile agar, commonly known as VRBA, as the selective media, which allows the coliforms to grow while inhibiting other type of bacteria. This medium contains crystal violet and bile salt, which inhibit the growth of most of the gram-positive bacteria. Neutral red is the dye and is included as a pH indicator. Lactose fermenting colonies like coliforms appear pink and may be surrounded by pink or red zones, indicating precipitation of the bile salt. So, material we are going to need are petri dishes, stomach bags, test tubes, pipette, cell spreader, ethanol, incubator, violet red bile agar, peptone water, and PBS. For sample preparation, measure about 10 grams of meat sample. Add the meat sample to 90 ml of peptone water in a stomacher bag or similar container. Thoroughly mix the meat and peptone water. Add 900 microliter of buffer peptone solution to 7 individual microcentrifuge tubes. Label the tubes starting from -2 to onwards. Add 100 microliter of meat and peptone water sample to the tube labeled as -2 and mix thoroughly. Take 100 microliter from -2, mix the sample and add it to next label tube -3. Repeat for the remaining tubes. Then for plating, add 100 microliter from the undiluted meat or peptone water sample to the first plate and spread the sample with a sterile spreader. Label it as -1. Repeat for the samples from each test tube, labeling each plate with the corresponding test tube number. Once the plates dried, turn them upside down before placing them into the incubator. This prevents the condensation from the dripping onto the agar. Incubate the plates at 37 degrees centigrade for the period of 24 hours. Here is a picture which shows pink colonies of Escherichia coli grown on VRBA. For recording the results, plate with 25 to 50 colonies should be used for the most accurate count of the colonies. If they are above 250, we'll consider it too numerous to count. For Salmonella test, oftentimes a harmful bacterium may be present on the meat sample, but in concentrations too low to detect by simply plating the resonate as in the previous test. These bacteria can be detected by first doing an enrichment step. Enrichment steps uses a selective media and is designed to allow the growth of target bacteria while inhibiting all or most of the other bacteria. This allows the target bacteria to grow to a higher and detectable levels. In case of Salmonella, the same principle can be applied to numerous types of bacteria simply by using different type of media. For this experiment, what we are going to need is petri dishes, plastic sample bags, test tubes, pipettes, cell spreader, ethanol, incubator, TT broth, tetrathionate broth medium, which is selective enrichment medium, RV broth, and XLT4 agar, which contain xylose, lysine, and tergitol. In the enrichment step, fill one stomacher bag with 90 ml of TT broth and one stomacher bag with 90 ml of RV broth. Add 10 gram of meat sample to each bag and mix thoroughly and loosely seal the bag. Incubate TT broth sample overnight at 37 degrees C and incubate RV sample overnight at 42 degrees centigrade. After that, enrichment stage comes the plating stage. After the overnight incubation, streak 10 microliter sample from each bag to individual XLT4 plates and incubate for overnight at 37 degrees centigrade. Salmonella positive samples will grow black colonies on XLT4. This is due to the fermentation of iron. Due to the enrichment step, as we have discussed earlier, only indicates whether the sample is positive or not. Additional methods such as most probable number can be used to estimate the quantity in the original sample prior to enrichment. So, here are some black colonies indicating iron fermentation. Typical Salmonella colonies will produce black colonies on XLT4. Some type of Escherichia coli or other coliforms may also grow on XLT4 medium, but not give black colonies. They usually produce high amount of acid, turning the medium yellow. As you can see here, yellowing of this medium is due to high amount of acids produced by Escherichia coli, and these black colonies indicate Salmonella colonies. Thank you so much.